Good afternoon, everyone. Welcome to this afternoon's session of the Wednesday afternoon lecture series here at the NIH. For those of you who don't know me, I'm Anirban Banerjee, a PI in the intramural program of NICHD, and also a coordinator of the Structural Biology Interest Group that nominated today's speaker. And it's really my great pleasure and honor to introduce Bill Clemons, who comes to us from the Division of Chemistry and Chemical Engineering at Caltech. In some way, we could call Bill as local because he received his undergraduate from Virginia Tech, where he got hooked onto biochemistry through his research on enzymes from Cryptococcus, an invasive fungal pathogen. And then he went on to do his PhD at the University of Utah in the then newly formed lab of Venki Ramakrishnan, where he was part of the team that solved the first structures of the ribosome. And this really led to ultimately Venki's being awarded the Nobel Prize in Chemistry in 2009. This is for the trainees. As a graduate student, Bill weathered the tedious task of freezing hundreds of crystals, one at a time in the cold room, by playing music. So for those of you who don't know, freezing crystals in the cold room with music, so we are in good company, as good as it can get. Bill went on to solve another giant challenge in structural biology in his postdoctoral research as a Demon-Runyon Cancer Research Fellow in the labs of Tom Rappaport and Steve Harrison at Harvard Medical School, that of the structure of the protein translocation channel. And subsequently, he joined the faculty of Caltech in 2005, rose to the ranks, and is currently the Arthur and Marion Hanisch Memorial Professor of Biochemistry. I'm pretty sure I pronounced that wrong. Research in Bill's lab has focused on several aspects, including how a class of membrane proteins known as tail-anchored proteins integrate into the membrane, and also protein glycosylation, which is a process by which sugars get added to proteins. Today he'll talk to us about how viruses that attack bacteria have developed simple protein-based mechanisms for breaking through the bacterial cell wall and killing the host. Besides all of this, Bill has been a tireless proponent of extending the reach of science to underserved communities. He has written about this, has spent a lot of time and efforts for this cause, and has very recently joined the Chan Zuckerberg science team, where he oversees a variety of funding programs, including the Science Diversity Leadership Award. And for that, we are collectively tremendously thankful to him for being such a role model of the citizen science. Today's CME code is 57665. I'll repeat that once again, 57665. For those of you watching on videocast, you can submit a question using the Send Live Feedback button, and your question will be relayed to the speaker at the end of the lecture. You can submit anytime. Thank you very much for coming to the NIH, Bill. Look forward to your seminar.
Okay, thank you, Anirban, and thank you all for coming, including those online. It's always a pleasure to be at NIH to see friends and talk science, and obviously the NIH has had a big impact on my life, both in terms of funding, but also serving in various roles to sort of help science move forward here too. And so anyways, I'm really grateful to have this opportunity to speak to you all. What I'm not grateful about is how cold it is. For this sort of a lecture, I normally would have worn a sports coat, but I was like, sweater seems like a better choice. So that's what you get. So today, the story I'm going to tell you about is titled Mechanisms of Phage-Derived Protein Antibiotics. And hopefully, if nothing else, by the end of that, you'll understand what that sentence means. And really, it's just a way to fight the growing problem of antimicrobial drug resistance. Now, the stories I'm going to tell you about really are driven by a few people. And so the first story, which is published now, is work that was started by a postdoc, the bulk of it was started by a postdoc, Nadia Riera, and then subsequently Karen Orta joined that project. She's now actually a former grad student as well, so they've both left the lab. And then Karen actually did a major part of the second story I'll tell you about. And then the third story, hopefully if we get there on time, we'll really talk about new work that's unpublished coming from a grad student, Evelyn Lee. So in all of my talks, I always start with a number 1619. I became aware of this number in sort of a real sense by the work of Nicole Hannah-Jones. She is amongst many things, she is a journalist who really took on this responsibility of understanding that in 2019, it was the 400th anniversary of this date. For those of you who don't recognize this number, she really put this out through the 1619 Project, where this date is the first documented selling of Africans to what would eventually become the American colonies. So the first enslaved people on our shores, not very far south from here in Virginia, happened in 1619. And so from that moment forward, through the Civil War, there were enslaved Africans on this continent. So slavery happened long before that. African slave trade started, pretty much almost immediately after Columbus discovered America. But this is really the sort of start of America, and the reason why, especially this week, following our holiday last week, I think is really interesting, because if we think about the story we're taught in school, which is also an apocryphal story, which doesn't make any sense in terms of historical context, and in fact I've learned recently that it has no historical basis on sort of even how Americans think about ourselves. But going back to Thanksgiving, when we talk about the Pilgrims and the Native Americans basically got together, the Wampanoag up in Massachusetts, greeted these Pilgrims to this meal. Apparently a meal really did happen, but it wasn't associated with Thanksgiving for many years. What happened to the Wampanoag pretty soon after this Thanksgiving was a pretty tragic event. All that said, we think of this as the start of America, and the Pilgrims, arrived on the Mayflower in 1620, right? So slavery was here before Pilgrims were here, right? And I think that's a really important thing to have in context when we think about the sort of state of America that we have today, where there's this real contrast in terms of wealth. The difference between black and white families, there's a tenfold difference in wealth in white versus black households. And all of that stems from history. We can document all of this, even though there are lots of people who would rather us not talk about it. There's a direct line between days like 1619 and today. And I just want to highlight the fact that there's a benefit to getting awarded to sort of special societies. And one of these was just a couple of months ago. I got to go meet at one of these events. I got to meet Nicole Hannah-Jones. This was the highlight of my academic career, and Shirley Malcolm, who's also works for AAAS and a good friend of mine. Okay, so when I was reflecting on this talk here today, there was something that I did think about in particular, and imagining sort of where we're at and sort of what the future might look like. There's a lot of discussion about racism and what does it mean for systems to be racist, and is it possible for systems to not talk about racism and there to be racism built into the system? And in fact, everybody in NIH knows these statistics that if you are black in America and applying for an NIH grant, you have a lower percentage chance. It used to be 10%. The last number was 8%. Hopefully it's lower now. But there is just statistically, you have a lower chance of getting an NIH grant just purely on nothing else but being black in America, right? Which is a crazy statistic, right? And you really can't separate that from anything but just pure racism built into the system. But there was this sort of interesting nexus point in American history. So at the end of the Civil War, during Reconstruction, federal troops moved into the South and basically tried to desegregate all of the different institutions. And one of the places where there was a real change in the sort of prospect for African Americans, black Americans, was in the federal workforce. And that was really by the fact that was the one place where you could say, look, we have tests, we have exams, you pass the exam, you can get the job. And this really became a major driving force for black middle class success up until the election of a certain president, Woodrow Wilson. Woodrow Wilson, for all of the things that he's well known for, I'm sure most people these days know the fact that Woodrow Wilson was also, he was a segregationist. He was a proud supporter of Jim Crow efforts. Woodrow Wilson, actually screened Birth of a Nation, which was a paean to the creation of the Ku Klux Klan in the White House. And so, it's not really hard to sort of understand, himself and sort of what he did in terms of founding things like League of Nations versus how he treated people in his own country. And this was actually, his policies. So he actually never actually said, we should remove black Americans from the federal workforce. But what he did say was that we should segregate black Americans from white Americans, and in doing so, effectively erased all of the success that black Americans had had getting into the middle class in the workforce. And he actually, when these policies were going into effect, members of the NAACP went to the White House and met with him, and he said this quote, segregation is not humiliating, but a benefit, and ought to be regarded by you gentlemen, which I think, is a good encapsulation of just the way that he saw the world. And, we can talk about lots of stories about sort of the effect of this and the long-term effect, and this goes even further into the future in terms of red line, in terms of lack of access to home loans, other things, lack of access to the GI Bill. But there's one story about this person, which there was a New York Times article written by his grandson. It tells a story about one of these workers, John Abraham Davis. And, he was an example of someone who he was born in D.C., basically spent his whole life in the D.C. area. His mother was a servant, working for a wealthy white family. His father turned out to be the lawyer who was the person in charge of that wealthy white family. He was never actually, his father never took ownership of him as a child, as his child, but his mom worked hard and he was able to get an education and become successful. There's a picture of him right here with his family. He actually was employed at the government printing office and actually had successfully rose into a position where he actually was in a position overseeing many white collar workers. And so he was, easily what we'd call middle class. He owned land. His family was well established. And when the Wilson administration came into power, they effectively took away everything that he had gained. So he basically went from 20 years of civil service to what he had achieved in terms of salary was cut in half. You can imagine going from, $150K a year to $75K a year. What would that do to your family? In his case, it probably wasn't even that sort of number. And they basically ended up losing everything. And so his family was reduced to poverty pretty quickly based on these policies. And his story is probably the best documented because he was very prolific in defending himself through letters. And so you can see all of that. But there are many of these cases. And in fact, the number is something like 30 to 50%. There's a 30 to 50% cut in salaries for black Americans working in the federal workforce. And so you can project all of that forward to today and sort of modern history and all these other things we can talk about. But I do think this is a sort of interesting time to sort of reflect on, what can an administration do, right? How can things change rather rapidly, and what are the long-term effects of what can happen? And I don't have any answers to sort of how we think about this, but I do think, as scientists, we have a tendency to not want to get engaged, and I don't know that the future can survive that. And so anyway, I think this is a useful thing to consider when we think about the history. I have been fortunate enough to be involved in a number of different projects. One of them is this paper that was really led by A.J. Hinton at Vanderbilt University. A.J. is a junior faculty member there, but he's really an amazing scientist and thought leader. And he really put together this paper. And I'm last author on this because for some reason he alphabetized it by first name, and so William apparently was last. I have no idea why he chose to do that. I contributed commentary and supported the publication, but I wasn't responsible for the writing. But this really is a great article that talks about, basically it was a cell issue reflecting on Juneteenth, a newly created national holiday, which I'm sure you all understand the history of that, hopefully by now, and if you don't, you should look it up. But it really talked about this sort of barriers to equitable science and sort of ways to overcome that. And so there's this nice graphic that really talks about sort of where the opportunities, where are the inequalities and how do we overcome them. And, as was already mentioned, I do also have this role at the Chan Zuckerberg Initiative, where my title is Program Officer for Diversity in Science. I get to do some cool things through that. This is one of our fireside chats where I'm getting to talk with Rick Kittles. He's the VP of Research at Morehouse Medical College. A brilliant guy, and he's been working hard to sort of transform their research program, and I learned a lot from him here. He's telling us about the importance to our society for these historically black medical colleges. But we have this saying at CZI, which is that diverse science is better science, and I think that's just a sort of fundamental thing to appreciate. We learn things. One of my colleagues there, she is a mentor of my team. I think many here probably know her, Hannah Valentine, and she told me the story about how in her field, a new class of cholesterol drugs that came out recently, these PCSK9 inhibitors, the discovery to create this novel drug that's useful for everybody really came from GWAS studies that were being done by Helen Hobbs and her colleagues to look at African genomes and basically identify this loss of function mutation in the cholesterol pathway. And that identification that identified a new drug target and then related this new drug to treat high cholesterol. And so, it's not just that these things benefit, studying diverse populations, understanding diverse populations, bringing diverse populations into the fold isn't just about, doing things because they're the right thing to do. It's also good for all of us from a discovery perspective. Okay, so I'll step off of that for a minute and now tell you a little about what my lab's been up to. We really divide ourselves into two camps in the lab. There's what I like to call the glycochemistry of biological membranes, where we like to think about the transfer of sugars onto and off of lipids and sort of the various mechanisms and proteins that are involved in doing that. And some of that will be the focus of today's talk with peptidoglycan biosynthesis. The other half of the lab is really interested in membrane protein biosynthesis, and there we focus on the special class of membrane proteins called tail anchor membrane proteins, which I don't have time to talk about today, so we won't get into it. But this is a really great team that I've been fortunate to work with over the years. All right. So one of the things that drives our work is this idea that of bacterial infections. The idea of plagues is not a new one, and it's also one that's very prescient on many people's minds. The reality is that disease is always right around the corner. And in fact, if you look at what life was like before antimicrobial drugs, people died of all kinds of things, right? Pneumonia killed lots of people every single year. It still kills people, but not at the same rates. But you can think of things that we don't even think about anymore. Diphtheria, although things like syphilis are on the rise again. It's a little disturbing where the world's at. But before we had any drugs, there were it was any injury that you had would lead to death, and things like 10% of skin infections resulted in the loss of a limb, right? If you can imagine that, if you're just walking through the woods, you get a scratch, and the next thing you know, an infection starts, and then you die, right? That's pretty much what you have, a limb amputated. And so, fortunately, without going into the history of all this, some of this, right? Alexander Fleming and others. But basically, antimicrobials were discovered. Penicillin became sort of the first sort of magic drug. And the problem is that antimicrobials are great. We all take them periodically. This is something that everybody uses. But resistance develops. And resistance develops actually, fairly rapidly. And so this is actually sort of a timeline really comparing just various classes of antimicrobials and then when clinical resistance was developed. And as you can see, for sort of the more modern drugs, resistance is seen in the clinic almost immediately. And so we now have this problem that there are many different bacterial strains that are completely resistant to all known antibiotics. And this, and it doesn't mean that there's no hope for small molecule. And my lab actually works on small molecule discovery, so we're interested in this. We have this really great new class of inhibitors called teixobactin, which I think is a pretty interesting compound. It targets lipid 2 in sort of this unique way and may actually work well. But this isn't enough. And if we don't figure out some sort of better mechanisms, the WHO estimates that between now and 2050, an estimated 39 million people will die from these so-called superbugs. These bugs are resistant to all known drugs. And if you do the math, that's one person every 20 seconds. And by 2050, as many people will die from bacterial infections every year as will die from cancer, right? This is a really, a big problem. And so what do we do? So we try to find new drugs, and we're working on that. The other thing you can do is turn to something new. I just found this quote I thought was really great. This is from Robert Schooley, who worked with Steph Strathi, who I'll mention in a minute, to really change the way we think about phage as a therapy. But he has this quote: "In phages, evolution has produced an effective killer of bacteria, one that offers enormous potential in the worldwide fight against antibiotic resistance." And I love that sort of connection about evolution, right? That we actually have things in nature that their sole aim in life is to reproduce by killing bacteria. And so we can learn a lot about this from them. And this really, if you go back in history, this isn't a new concept. So back in the early 1900s, Twort and de Harel separately discovered bacteriophages. And Felix de Harel was really amazing because he immediately recognized that you could leverage bacteriophages as a therapy. He started an institute in Paris, really to find phages. He was curing people of dysentery using bacteriophage. Eventually, some of the people that he'd interacted with eventually set up labs other places. One of the most famous of these is the Eliava Institute, now the Eliava Institute in Georgia, in Tbilisi. But what was interesting is this all was happening at the same time that penicillin became rapidly available. And the discovery of penicillin basically led to the birth of the pharmaceutical industry, and suddenly there was no interest in trying to develop bacteriophage, at least in the West. About the same time, Stalin had risen to power, and World War I happened. We're heading into World War II. Things fell behind the Iron Curtain and basically separated sort of the sciences. And phage research actually still continues to this day in former Soviet countries, including in Georgia. And in fact, if you go to a former Soviet country or even things in their sphere, you can go to the pharmacy and they will give you phages for internal bacterial infections. It's pretty amazing that this is already a therapy in parts of the world. We don't have access to this ourselves. And it's somewhat questionable how this actually works all the time, but it certainly, now that I've been telling this story, I've heard from several people who say, yeah, I took it. It was really great. It cured me, right? So, that's interesting, right? This isn't new, but it is something that we haven't thought about in the West for a long time. And really the driver for the changes in the way you think about phage therapy really came by this story from Steph Drafthy and her husband Tom Patterson. And, just for brevity, you should read this book if you're looking for a good book to read. This is a good one. But it really describes the fact that Tom and Steph were on vacation. He had a health complication. He went to a hospital and there contracted a bacterial infection that eventually became systemic and was incurable by any of the known drugs. And so Steph, because she was an epidemiologist and had connections pretty broadly in the sort of health sciences community on both basic science and medicine, was actually able to get phage therapy for her husband and cured him of his bacterial infection. So this is the first example of a patient cured by bacteriophage in what we call the West. And this was a huge deal because they had to overcome FDA regulatory problems that had been huge problems keeping phage therapy from becoming something. And eventually they actually founded this institute. So both Dr. Schooley and Dr. Strathy are the co-heads of this Center for Innovative Phage Therapeutics at UCSD. And here they're actually curing patients, and so this is pretty exciting. And this isn't novel. There's other centers around the world that are doing this. There's a huge number of scientists now who are really committed to this. And in fact, startups have started to form. And so this is just showing the number of startups the number of trials being submitted by startups based on phages. And you see basically around the time when this story got published, you see sort of this ramping up now of trials. And so I think, phage therapy is here to stay. All right, so we're going to get back to phage in a minute, but there's other things you need to know. And one of those things for this talk is to think about bacteria. So we're really interested in targeting bacteria. This is an illustration from David Goodsell of E. coli. And the part that we're interested in, isn't the bacteria itself, but it's really the cell wall. And so we can zoom in and sort of have a look. So he's got this more high-resolution picture that's really based on structures. And you can see, kind of things that probably stand out to you. Not that even if you don't look at bacteria all the time, you can see this is the genome here. This is one of my favorite molecules, the ribosome here, just sort of making proteins. And the part that we're most interested in is this part here, the cell wall. Now, the constituents of the cell wall involve, membranes and sugars. This is E. coli is gram-negative. That means it has two membranes. And the cytoplasmic membrane would be this one on the inside here. This sort of encloses the cytoplasm. Embedded in this membrane are membrane proteins. I'm just showing this just to remind you how to look at membrane proteins. And so if you're not familiar with how to think about membrane proteins, the thing that's important about membrane proteins is that they're embedded in these lipid bilayers. So they have this very hydrophobic surface where they basically have to live in this unique environment. We're interested in them because, like all proteins, are made on ribosomes, and they have to get into the membrane and fold there. And so there's lots of still big questions to answer about how that works. And so we're interested in that in my group. But it's also important to sort of note that that these membranes are really these sort of dynamic environments. And so other things to sort of see here, it's a protein, it's a protein structure, it's folded up. These are actually alpha helices these are helices, transmembrane domains as we refer to them, that cross the membrane. And so in this case, there are 6 transmembrane domains. And so you're going to see a lot of membrane protein structure today. And so just to give you some framing how to look on it. The real sort of centerpiece for today's talk in terms of biology is this layer here between the 2 membranes in gram-negative bacteria. In gram-positive, this will be exposed on the surface of the cell, but this is a layer called peptidoglycan. And peptidoglycan is really an interesting molecule because peptidoglycan has this feature that its role in the cell is actually maintain the sort of strength of the surface of the cell. You can think of it almost like the exoskeleton or chainmail armor that surrounds the cell. It's a single molecule, so that's kind of cool from a chemistry perspective. It's this sort of massive polymer, so much so that you can boil cells in SDS, and you actually the only thing that survives that is the peptidoglycan, which you can put on an EM grid and have a look at it. We know a lot about how this is made, and so the process really starts in the cytoplasm. And so there's a series of enzymes that work to actually make a special UDP sugar pentapeptide, as sort of the key soluble unit. This phosphosugar pentapeptide gets transferred onto a phospho, what's called bactoprenol or polyisoprenyl chain, to this phospholipid to generate a diphosphasugar pentapeptide lipid, which is referred to as lipid 1. The enzyme that catalyzes this is MraY, and it catalyzes this sort of interesting reaction. We have these sort of 2 completely different kinds of components. One large soluble component where this phospho group is going to get transferred onto this phospholipid, which is long-chain polyisoprenyl lipid. Once this has been made, lipid 1's been made, a second enzyme comes in to catalyze, transfer another sugar to get lipid 2. So now you've got a disaccharide here. This is actually pretty bulky, so to get across the membrane, it has to be flipped by this enzyme, by this flippase called MurJ, and then a series of enzymes basically stitch this together to make this polymer of sugars that are cross-linked intermittently by amino acids, and that's what makes peptidoglycan. Now, of course, we're talking about this because this is a really great target for drugs, and in fact, the first drug, penicillin, targets this. There's lots of points in this pathway that are targeted by clinical drugs that we use today, and so this is just a small sampling of them, but we basically hit a number of these pathways. But one of the things you'll note is that we, as of right now, don't have any clinical drugs that target any of the membrane steps. Now, that said, we do know that there are natural products that have been developed that have been studied that do target these steps, and so my lab's interested in understanding the mechanism of these and leveraging these. In some cases, we had some success with our synthetic colleagues in developing these as potential therapeutics. But for today's talk, what we're most interested in is that a number of these steps in the pathway have been targeted by phages. All right, and so the 3 phages that I'll show you structures for today, well, the 3 targets I'll show you structures for today, Phage X, which targets MraY, Phage M, which targets MurJ, and Q beta, which targets MurA. All right, so back to bacteriophage. What are bacteriophages? They're viruses. Like all viruses, their life cycle means they have to find their host. All right, their life cycle is they have to find their host. They have to then insert their DNA. And for these large bacteriophages, they insert their genomes. They take over the host. They basically destroy the bacteria itself, make lots of copies of itself, and then have this problem where once they're fully assembled, they have to get out. And so that's a key step, is how do you get out of the cell? If you don't have a mechanism for breaking through the cell wall, you're going to be trapped, and that's going to be a problem. You're just going to be you're going to be a peptidoglycan bag full of phage particles. And so this is the key step that we're actually most interested in. And this phage that I'm showing here is sort of, it's a cartoon version of a T4 phage. But if you look at the T4 genome, it's actually quite large, 160 kb. And so these phage genomes actually are fairly large. They do a lot of things, and they can encode for a lot of information. So they actually have fairly complex mechanisms for going through that cell lysis project. The phages we're interested in are actually not part of these large phages. They're actually small phages. And sort of a key player in the history of this project that we worked on, at least this first half, really came from Bob Sinsheimer. He was a Caltech faculty member, and he wrote this article talking about his phage of interest, which was Phage X174, in a book that was put together in honor of Max Delbruck's 60th birthday, where he put together this essay titled Phage X, Multum and Parvo, a lot and a little. And so he was a physicist who'd gotten into biology, and he wanted to study phage. He thought phage could tell us a lot about life, and in fact it has. But he picked Phage X because it was the smallest genome that he could find, the smallest phage that he could find. And he thought that would be the easiest one to work on. And in fact, he was right. This one became really important to biology. The Phage X174 had many firsts. It's thought that this was probably in the first phage cocktail that was used by Bacterial back in the 1920s. But it was the first example of a single-stranded DNA, first example of a circular DNA, first example of a synthesized self-replicating DNA. So this was Roger Kornberg's work, but they basically showed that you can make copies of this in vitro and then still be infectious. It was the first full identification of all the genes. Geneticists are pretty awesome, and they figured this out using phage genetics. It eventually was the first restriction map that was actually established. Probably the thing most people who came of age in the 1970s know it for, though, is this is the first sequenced DNA genome. And in fact, it was sequencing this genome that won Fred Sanger a second Nobel Prize. It was also the first synthetic genome ever made. And so this was at the Ventnor Institute actually synthesized the genome purely from chemistry, which was a pretty cool result. Okay, so, I got ahead of myself there. All right, so what is Phage X174? So Phage X174, these are two different kinds. The phage itself, this is the active phage in solution, but in the bacteria, it starts off as a prophage with this green protein that surrounds it. That's protein D. Its genome is about 5.4 kb, which is tiny by any stretch, and so it can encode very much information. Even at the structural level here, these structural genes that you identify, you can sort of map them all onto it. But our gene of interest is really a different gene here, which is this gene E, lysis gene E, which encodes for something that we call protein E. And we're going to talk a lot about this protein E for the next 10 minutes, but the thing that's one of the things that's interesting about it, which I won't talk anymore about it, is the fact that it's a nested gene. So this is the first example of a gene that's of a stretch of DNA that encodes two different proteins. And so it's in a +1 reading frame relative to this D gene, and therefore any sort of evolution can't really optimize this gene without the consequence of making the other gene worse, right? So there has to be sort of co-evolution for these to improve, which is an interesting principle associated with this lysis gene, and turns out to be a kind of common thing you see for the ones I'll tell you about. Okay, so when we talk about mechanisms of lysis by a bacteriophage, this is a very simplified version, but T4 phage is something like this, where at some point under EM, you basically see that this sort of catastrophic explosion of the cell, and all of the bacteriophages spill out. And they do this by encoding several different proteins that work in contrast. You have endolysins, which chew up the glycan. You have holins, which punch holes in the cytoplasmic membrane and release those endolysins. And then you have these proteins called spanins, which couple that breaking of the inner membrane and the peptidoglycan with lysing of the outer membrane. These proteins all work in tandem to sort of release the cell in a very well-timed mechanism. For the phages that we're interested in, like Phage X174, which are these small single-stranded nucleotide phages, their mechanisms are much simpler. And this was first appreciated very early on, was that Phage X itself actually had a lysis profile where the phenotype looked very similar to what the phenotype was for penicillin, implying that what it was doing was something similar to inhibiting peptidoglycan biogenesis. And in fact, in all of these cases, it's a single gene that does this mechanism for lysis. And so the person who really turned us on to this whole field, MraY coined this concept, single gene lysis, or sigils, as we call them. Phage X, this is just a different coloring scheme here, but this is gene E, or protein E, from Phage X, which is the lysis gene for this. These sigils are defined as protein antibiotics that are sufficient for killing bacteria when expressed in the bacteria. So all by themselves, you can kill the bacteria. There are lots of examples of these, and these are just sort of the major ones that we'll sort of talk about today. The other ones are all single-stranded RNA genomes. You'll notice they're actually much smaller, so on average maybe around 4 kb. In these cases, they really only encode for 4 clear genes that are encoded by each one of these phages. So that's all they need to do all the things that a phage needs to do, and that's, I think, pretty cool. But the other thing you'll note is, though, even though these are all leviviruses, so they're all related to each other, they actually have very different mechanisms for lysis. They have different kinds of genes that show up all over the genome. One of the sort of more interesting ones to just talk about first is the first one. We understood the mechanism at a structural level, which was work that MraY had actually done with a collaborator on the Zhang lab at Texas A&M, where they actually were able to show that this protein Q beta, which is a maturase gene. It has multiple roles, including binding to the receptor on the bacterial cell, but it also sequesters the first enzyme in the pathway, MurJ. And so it basically works by just taking this, starting with the first enzyme and taking the pathway out by just binding. All the phage particles would be decorated with these MurJs, and then you cannot make peptidoglycan. And so that's a pretty interesting mechanism. Now, the one we're talking about next is this Phage X174. It expresses this protein E. The Phage protein E has this sort of interesting structure where it's got an N-terminal transmembrane domain followed by a soluble C-terminal domain. About 91 amino acids, and we'll look at that in more detail. So the first insight into what mechanism for what's working with Phage, this protein E and how it worked really came out of MraY Young's lab. Tom Bernhardt, he's at Harvard now, but he was a grad student with MraY Young, and actually I should say Harvard Medical School now, so Tom's a professor of bacteriology at Harvard Med. But he was working with MraY Young as a PhD student, and together with others in the group, they really established explicitly what the mechanism for E was. And they made a number of different discoveries. One was that this protein E was dependent on a cytoplasmic chaperone called slide E, which is interesting for a variety of reasons. I'm not going to talk in any detail about it today. But it's a non-essential gene. So you actually can knock slide E out, and you can actually then evolve Phage X that could target this. And so wild-type Phage X requires slide E, but it's not essential for the mechanism for lysis. It's not an essential gene. Based on that, they're eventually able to identify mutations in MraY that would show resistance to this Phage X-mediated lysis. And so MraY now is sort of a central player, right? This is a key central protein in this enzymatic pathway, the first membrane step of peptidoglycan biogenesis. All right, so we can talk about MraY structure. Let's look at the parts that we have right now. So the first structure, MraY, was solved by the Lee lab at Duke from Aquifex aeolicus. It's a homodimer, so we call it a back-to-back homodimer, because these two angled helices really define a groove here where the active site is. And so the lipid substrate comes in for the plane of the membrane, the soluble substrate comes in here, and then MraY catalyzes that transfer, that phospho sugar onto the phospholipid to generate lipid 2. The other players, so again, we have this protein E from Phage X174. It turns out these single-stranded DNA phages, there's lots of sort of variants. They're all pretty much the same to my eyes, but the virologists like to give all these different names to these things. But they have all of these consistent features. One is this virtually completely conserved transmembrane domain, and then these conserved, in the cytoplasmic domain, these conserved positive these conserved positively charged residues here, lysines and arginines. And the one thing I want to draw your eye to is that there's this completely conserved proline, which is predicted to be in the middle of the membrane. And so this was really interesting for a variety of reasons, but it's completely conserved and actually essential for lysis. The other player is this slide. We also saw structures from two different versions of this. They look virtually the same, so we won't go into that in any more detail. Just to mention that we do have two different strains of these. The other protein is a proteolysomerase chaperone slide E. Slide E is, again, interesting for a variety of reasons. It has this FWP domain, this sort of clear proteolysomerization domain. And then it has what the original authors who discovered this called this insert and flap domain. And these two domains, this basically binds to extended peptides. This binds and catalyzes the interconversion of prolines from cis to trans. And these two domains work together to actually be really efficient chaperones for folding in vitro. But what you can see is even though they're well-folded domains, they're actually pretty flexible relative to each other, which introduces its own problems from a structural biology perspective. So our work in this really started when a postdoc, Shio Tanaka, took this on. I basically presented this idea to her and said, look, my guess is that protein E and MraY are going to form a stable complex. So why don't you put a tag on protein E and see if you can purify MraY. So that's what she did. And so she put a tag on protein E, co-expressed it with MraY, and then pulled out this complex that contained these two other bands that turned out to both be E. coli slide E. Right, so this is the native slide E. So in this particular gel, this was the first experiment we did for this. We didn't actually expect to be another band there, and it turns out it was. So eventually, the two bands really relate to the fact that slide E has this long, this flexible C-terminus. It has a lot of cysteines and histidines on there, and if you delete them, you only see a single band. But we got a beautiful trace on a gel filtration column. We got crystals, and basically for 8 years tried to get those crystals to diffract, and never got a structure. And then Cryo-EM kind of changed everything. So we finally, and so I should say we call this the YES complex for MraY, protein E, and slide E, so the three-component complex. And YES sounds cool, so the YES complex. This is my student Karen. I'm actually loading some YES complex grids into our microscope. And you get data that looks like this. I still don't understand why this works. I mean, I understand the principle, but you get these sort of fuzzy images. And you feed those in the computer, and they eventually give you these 2D projections. And you basically take these particles, and eventually, just through complex processing and computationally, you can get to high resolution, which is a subset of those particles. And so our eventual maps, which I'm going to show you next, were about 3.3 angstrom resolution. So the YES complex itself, this is just showing the YES complex. Now it's in my cell right now, and so we're contoured at a level where we can see the disordered regions. But of course, the most interesting thing is to sort of see, where the protein is. And so now we can actually we can contour it down to remove this. And of course, all structural biologists in the audience go, oh, those TMs look great. And this is exciting, right? So when we first saw this, we were obviously really excited. Now, now we know what MraY looks like, right? There's a structure of that. We know what slide E looks like, so we can actually identify those parts of the density. And the only other part of the density that we see here is the yellow density, which is going to be now this viral density. So now this is this viral density that comes in and interacts with this complex. And the density is quite good, so we can actually build this in at high resolution and get an atomic model. And so we can basically replace that model with something that looks like this. And the thing there's a lot of things to take away from this, and just for time, this is all published, so I don't want to get in a lot of detail for this, except to say that when you look at the structure here, what you can see is this is an E. coli protein. These are E. coli proteins. They have zero interest in each other. It's only through this interaction of the phage that these proteins come together. So this phage protein is expressed and brings these together. Now, what is slide E doing? It's not actually doing anything particularly important to the actual mechanism of how blocking lysis happens. What it does is it grabs onto these C-terminuses of slide E in this sort of bow tie kind of interaction. And we believe that the role for that is really to stabilize this complex, to make it essentially an irreversible complex. And so that's the concept that goes there. Other things to point out, well, let's go to the next slide. So how does that actually work? The mechanism is not super hard to imagine. You've got this transmembrane domain for slide E, for protein E, that basically lines the groove where the active site is, and it prevents access of the lipid substrate to that active site. So there's not a whole lot of complexity there in understanding how it works. You basically block the active site, and you're no longer able to catalyze synthesis of lipid 1. It just acts like an antibiotic. The other thing to note here is I pointed out there was this completely conserved proline in the transmembrane domain. And that proline is right here. We see this kink in slide E. And so this proline, the role for this proline, we presume the role for this proline is really to put this kink, which is not something that a transmembrane domain wants to do. You put this kink in there and it allows it to sort of follow the shape of this active site, to sort of bind tightly in the active site. And in fact, these other two prolines give very strong phenotypes as well. And so that shape complementarity of that groove is really essential. With this, we can actually map all the resistance mutants, and so kind of just to wrap this story up, we actually see all the resistance mutants to protein E all map to this interface between protein E and MraY. So that's what you would expect. Okay, so the mechanism is really simple, right? The phage expresses this peptide. This peptide binds to the complex, stabilized by slide E, and now you can no longer get lipid 1 synthesis. And so now we have our second sigil. So the other thing that's sort of notable about our structure is actually the first cryo-EM structure of MraY. It's the first structure of MraY from E. coli. And so we can actually say a lot about function, and we talk a little bit about this in the manuscript. But one of the things that we could see here, because this is in cryo-EM, we actually could see a lot of density for lipids. Now, we didn't really make anything out of these lipids in the manuscript because we couldn't say very much. But one of the things that we were kind of surprised by is that we saw this long continuous density here, which is like, that's kind of weird. Doesn't look like a sort of typical phospholipid. But in the writing of this, as we're finishing up this story, a paper came out from Carol Robinson's group, where they'd done a mass spec study and identified that it seemed like MraY had a C55P binding site. And then she collaborated with Robin Corey and Phil Stansfield at the University of Warwick to do MD simulations. And they actually localized a head group for a C55P lipid right where this density ends. And they identified that these residues are sort of key to that, and this is right what we see in our density. So we don't know anything more than that to say that was an observation that we made. But it seems plausible. The other thing was that if you look at the center of this complex, we actually saw that between the two dimers, it's filled with this density here. And we didn't know what to make of this density at the time. Well, we had some theories. But it's interesting. So actually in all MRAY structures, there's a cavity there at the dimer interface. And ours was probably the most, the best density that you could see, but we still couldn't tell what it was, except for the fact that it had this sort of interesting sort of curly-Q density there. And we thought, what's that telling us? And really, it was talking to my friend Tom Bernhardt and his postdoc, Lindsay Marmont, which she was telling us some of the work that she was doing that gave us, I think, the best insight that we both came to the same conclusion. And so just to tell the story rather quickly, Lindsay was interested in the fact that they developed a strain of bacteria where they were looking at these penicillin-binding proteins. So these proteins are responsible for basically stitching together peptidoglycan. These proteins in gram-negative bacteria require activators. So each of these, there's two different PBPs in E. coli, or in this case, it's Pseudomonas aeruginosa. There's two different PBPs. They both have their two independent activators. And so for this particular study, they knocked out one of the activators, they knocked out one of the PBPs, and now they have a very low production of peptidoglycan. And so you think, okay, that's now these cells become sensitive to different kinds of growth conditions, and so they could look for mutations that actually recover viability in the presence of these knockouts under these stress conditions. And the thing that they discovered was a mutation in MraY called T23P, which didn't really make any sense that this would be an MraY until you sort of think of the full picture. And so based on that, they looked for other mutations in MraY, and what they discovered was that all of their mutations really mapped to the dimer interface in this groove that we see between the two MraY monomers. And so that was really interesting, and it suggested that groove was actually playing an important role in the function of MraY. And Lindsay went on to show that mutation, so this T23 mutation, actually creates a hyperactive MraY. And so you make this mutation, and you suddenly see, and this basically, these are two different ways of looking at this, but the short answer is that this is Pseudomonas and E. coli. In both cases, you make this mutation. The wild type, if you express the wild type protein, you basically see not a very low increase in the total amount of lipid 2 that's produced. This is all in the bacteria, and then you basically purify the lipid 2, and then you look at it directly. Whereas in the mutant, you see this huge upregulation of this, right? And so it's like, okay, clearly it's an activating mutation, but why would it be activating? So when we then thought about this from the perspective of our density, our density is an inhibited complex. And so we thought, maybe what we're looking at here is an inhibited complex here. And maybe the density that we're seeing here is actually some sort of an inhibitory complex that might be true. Now, we couldn't get this to a resolution where we could resolve what was going on there. This is improved density from our previously published work. But again, like a lot of folks in this field, turned to Phil Stansfeld at Warwick and his team, and they basically did some MD to really ask this question. And so one of the things that they could show is sort of in coarse grain MD, lipid 2 happily goes right in the middle of that cavity. Other lipids don't go there. So only lipid 1 and lipid 2 go there, except that this is now on the periplasmic side, so there's only going to be lipid 2. And if you actually, do full atomistic simulation and then look at sort of the position there, you basically see that lipid 2, it occupies the groove pretty happily there. And so the model that really derives from this is a pretty simple one. The idea here is the pathway, if everything's flowing along fine, MraY is happily catalyzing lipid 1, converting that to lipid 2, you flip that across the membrane, and then you make peptidoglycan. Now, the problem is if MraY is chugging along really fast, but making a peptidoglycan is going really slow, you're going to get an accumulation now of lipid 1 and lipid 2 especially. Lipid 2 buildup is actually toxic to the cell for a variety of reasons. But to prevent that lipid 2 buildup, the thing you're going to do is allow lipid 2 to come back and inhibit MraY, and you basically shut the process down here. Now, the reason it's toxic, and Lindsay was able to show this clearly, was that this precursor, the C55P, the polyisoprenyl phosphate, is essential to a number of different pathways. And so basically every sugar that has to get to the outside of the cell finds its way there based on this carrier protein, this carrier lipid. And so if you shut down, if you focus everything on one pathway, all the other pathways suffer for it. And we noticed actually, if you compare the previously published crystal structures from the inhibited state and the apo state, there's only one apo state that's published so far. And the thing that we noted there was if you compare one monomer to the sort of where the position of the other monomer is, there's actually an opening and closing that we see. So we don't know more of it than that, except to say that all of this is very plausible as a mechanism. That may be true for this general class of inhibitors. Okay, so just to to get back to our phage question. So I already told you about these two sigil mechanisms. One of the things that was published recently from MraY Young's group, his grad student Frankie Antolon, was this mechanism for actually looking at these sigils directly. And so the idea here is we actually can find sigils all over the place. Can we identify how they work mechanistically? And so they developed this assay where basically in cells, if you overexpress any of the sigils I've been telling you about, you get this phenotype here where during septal division you get this blebbing and the cells basically break. And so it's very clean. You can see this always at the septal division point. You see this. But you actually can look at this directly in the cells. And so what you see for these is this, just to walk you through this. So this trace here is just measuring OD. And so what you see is as cells are growing, you induce, cells rapidly start to die based on the fact that you've inhibited peptidoglycan. Before they start to die, you actually can monitor the incorporation of peptidoglycan into the cell wall. And what you can see is that with these inhibitors, you suddenly see loss of incorporation of peptidoglycan, suggesting that it's inhibiting this pathway. And there's lots of controls to support this. And this is all the sigils I've told you about it, but it turns out there's another class of sigils that we call type 2. They call type 2, so now we call them type 2. And in these cases, they all kill cells similarly, except in these cases, you don't get lysis at the septal division point, and they don't inhibit peptidoglycan biogenesis. And so we don't know what these are doing. Of the ones that I'm showing here, the best study of these is MS2. We still, to this day, have been studying for 30 years. We have no idea what its mechanism is. And so these are sort of exciting questions because there's exciting biology to learn from this. But the other two that are on here are both also type 1s. And so I'm going to show you those briefly, and then I'll wrap up. Okay, so I'd already mentioned that one of those targets MurJ, and it turns out, as we'll see in a moment, they both target MurJ. And to remind you, basically, MurJ is a sort of flippase, which is responsible for this sort of last step in flipping this disaccharopentapeptide across the membrane. And the flippase MurJ is part of this MOP family of transporters, and it basically has this inward-outward conformational change where you just alter accessibility, and the head group goes in on one side, and then you alter accessibility, comes out the other side. And so we still don't understand the details of this mechanism, but there's a number of structures, most of these coming from the Lee lab, again, at Duke, that really map these out. So in the last couple of years, the first result that came out was about Phage M. So it was a paper that came out from the Young lab where they basically established that the peptide that's expressed, 37 amino acids alone, directly targets this MurJ protein. Okay, so Phage M is the first, 37 amino acids. Last year, another paper came out looking at a Pseudomonas phage, PP7. Again, this is all done in E. coli, so that you see the same in E. coli. And they basically identified that this one, 75 amino acids, again with a single transmembrane domain, also targets M. And for time, I won't go through the data except to say that they found, or MurJ. So we have two different sigils that target MurJ, sigil M and sigil PP7. They both come from different parts of these liver virus genomes, and so even here you can say that they're convergently evolved to target this thing. So what do they look like? So the way we got structures for this was really straightforward, and we took advantage of the fact that Andy Cruz at Harvard had solved a crystal structure of E. coli MurJ where he put a Brill domain on it. This is the 4 helical bundle that allowed that to crystallize. For us, this was really useful because we basically took this, we made our own version of this, but we took this construct and we could show that we could purify this stably with sigil M, and we did some controls to show that it would rescue lysis. But then we have the benefit that Tony Kostyakov had actually generated, at University of Chicago, had generated antibodies that would, Fabs that would bind to the Brill domain, and actually a stabilizing antibody that binds to the Fab. And so we can basically add those into our complex. And then right from the start, from our first reconstructions, we could clearly see our MurJ proteins. And the reason we did this was MurJ is pretty small, and so we needed to add these other pieces. And so we could, the Brill, and the antibody, and then there's our protein, the transmembrane domains. And right from the start at low resolution, we could see this density that was bound there. So, by just focusing on the membrane port, we can get to high resolution. So we have a 3.5 angstrom map of this now. And so here's the map, and you can see this is where this sigil M is bound. If we look at where it localizes, sort of comparing to these Thermosyphilis africanus structures, it really seems to bind to this outward-facing conformation. It doesn't seem. It binds to the outward-facing conformation, which actually correlates with all of the mutants. So all the mutants sort of line the interface here and this outward-facing conformation, which was the prediction that it would bind this outward-facing conformation. And the idea is really simple. It basically just binds to this and locks it in a conformation where it can't convert between the inward and outward-facing conformation. Okay, so that's one of these. So what is this other peptide doing? So it's PP7, sigil PP7. It turns out it's doing something very similar. So just like M, it locks it in an outward-facing conformation. It's got its sort of transmembrane domain, which sort of locks it at this interface. The big difference is that unlike sigil M, which kind of goes in and comes out, sigil PP7 comes in and then goes down into the binding site for the lipid head group. And that's really interesting, right? It basically goes in and sort of occupies this space down here in the central cavity here. And so all that said, in both cases, they do something very similar. They lock it in a conformation where it can't go through these conformational changes. So, and so if we compare these two, you can see that there's differences, but at the sequence level, there's no relationship between these. And at the functional level, they basically do something similar. And what they've really done is told us there's a very key hotspot for targeting MurJ, which is super attractive because it's an outward-facing conformation, which means it's completely accessible to lots of different kinds of small molecules, or even cyclic peptides or something along those ways. And so we're thinking about that quite a lot these days. And so we have these structures now, but of course the sort of gates are sort of wide open, right? We can start to fill this in. So of these ones I have here, we now have structures of all of the known ones where we know the mechanism. But this is really just tip of the iceberg because it turns out there's thousands of levivirus genomes that we can look at. And in fact, we're all full of leviviruses. So it'd be great if I had the ability to culture all of your, or sequence all of your leviviruses, because we're almost certain to find novel sigils here. And even in these cases, right, these are 3 different ways that phages have targeted enzymes in this pathway, but it's undoubtedly going to be more mechanisms of this. MurJ seems to be a hotspot. MurJ is a flippase. There aren't examples like this of sort of targeting flippases. So I think this MurJ is really cool for a variety of reasons. I expect we'll find these again, but we'll probably find other members of the pathway, and we'll probably even find novel ways to target the bacteria by leveraging these. And so this is sort of the ongoing work in the lab. All right, so just to sort of conclude, so I told you today about the mechanism protein E and how Phage X174 establishes the S complex as a mechanism for killing bacteria. Talked a little bit about this sort of interesting property of these enzymes that sort of came out of this sort of structural and genetic analysis, which really suggests that there is a sort of very logical feedback inhibition mechanism for this pathway, which will be fun to explore and also maybe a useful target for thinking about therapeutics as well, because if you can shut down MRAY from the periplasm, that could be great. And then finally, I told you about these convergently evolved SGLs. And the convergently evolved is really interesting because in all these cases, these are completely new sequences that nature just discovers. And so it suggests that we could actually do this ourselves through different mechanisms of evolution in the lab, but we're going to be able to find all kinds of ways to kill. Probably every single way you can imagine killing a bacteria can be found using these kinds of phages, at least by a peptide. All right, so I'll stop there and just say, I mentioned most of the folks in bold here who did all the work and the collaborators who support us with this. I didn't get a chance to talk about our work with the Carosi lab at the University of Tennessee, but he's a synthetic chemist who's really helped us through all of our MRY inhibitors. And I will stop there, and thank you for your attention, and be happy to take any questions.
Please use no questions online just yet. If someone has, you can use the mics on either side. Looks like there's going to be a bunch.
So I was struck with your protein E, how it's interacting with these 2 proteins, and I just wondered, what are you thinking about sort of the order in which the proteins are coming together? Does protein E go into the membrane first and then drag the slide D into the membrane? Or I just am wondering how you're thinking about it, because a large surface is covered by those 2 proteins.
Yeah, no, it's a great question, and I really have no clean answer for it. What I do know, I can share, we talked about this in the paper, but one of the things we know is that if you look at this, where's my mouse? If you look at this, at the extreme C-terminus here, there's some prolines there, but they're not all conserved in the exact position, but there's almost always a proline here. We know that at least in the ones we've looked at, if you mutate that, there's a pretty significant phenotype. Now, that proline shouldn't make, once you make this complex, it's not essential. We can form the complex in the absence of the prolyl isomerization. And so we know that that's not critical. And so I think it's probably has something to do with what you're talking about. That probably what happens is you get protein E in the membrane. Slide E is the next thing to come in because it finds this proline and just kind of holds on to it long enough for the whole thing to come and collapse onto MraY. But, you know, that's all speculation.
So can I follow up on that?
Sure.
In terms of uninfected cells, what's known about what slide D is recognizing, and is there anything sort of akin to this proline rich structure that you were mentioning?
You know, I was talking I gave a talk, or I was visiting somewhere recently where somebody actually had worked on slide E quite extensively, and they were pretty excited about all the things that our structure gave them because no one's really looked at something that slide E actively binds to. But it's not essential, but it is actually an important protein. So it's, you know, almost all it's not gram-positive bacteria, almost, I don't know of any gram-positive bacteria that have a slide E, but almost all gram-negative that I know of have a slide E. And so it definitely is an important protein. It seems to be really key in things like the TAT pathway for secretion of metalloproteins. And so that, it's got this, in E. coli, it's got this long tail, as I mentioned, with cysteines and histidines. Those all, you imagine right away that must be involved in metal binding. And it seems to act as a chaperone for those proteins to really, stabilize their purpose. But, I don't really know more than that.
Thank you.
Yeah, it's constitutionally expressed as well. I mean, that's really cool. Harris.
Yeah, hi, Bill. That was a great talk. Just another question about protein E, which is an intermembrane protein made in the cytoplasm. So do you have any thoughts about how you can use what you've learned about protein E and its interaction with MraY to develop a real-world antibiotic that would come in from the outside of the cell?
Yeah, MraY, the protein E is for all the reasons you're suggesting, is a less attractive mechanism. I think we're more excited about MurJ and the possibility, and this cavity, I think, on the periplasmic side, also I think could be really interesting. I think from this, it depends on what you think of as a useful therapeutic. So one of the things we know is that for every single one of these sigils, we can always make them better. So even just, myself and students staring at it, we can go, okay, we make this mutation here. They always lyse more efficiently. Better is a different definition, but we can get them to lyse more efficiently, which suggests they're more stably bound. And so I think, if you're trying to engineer cell death in some way in a bacteria, there's other ways to do it. But I think these are just really trivial things, right? You make this small peptide, if you put it in tandem with something that gets activated in the presence of some other signal, and then kills. I like to think of it as sort of, probiotics or something along those lines, right? Where if you wanted something that would, selectively knock the bacteria knock itself out, this is a great way to engineer that. But for a drug, it does suggest that if you could get anything that could get in there and sort of block that lipid access site, that would be great. None of the known drugs that we have, none of the natural products we have right now do that. How you design that, that's a whole different question.
Bill, that was fantastic. Sort of one question, perhaps two parts. So your protein E sort of engaging the two proteins that otherwise don't care for each other, it reminds me of this small molecule borrowing a protein surface where you have like FKBP and FK506, and then the small molecule binds and it brings a second protein. So do you think that this could be a general mechanism through which you can essentially sort of either design a protein that would then bring another protein, sort of akin to what people are doing for PROTACs. So essentially regulating protein function by sort of, adding something and that then, drags something else from the cell. And so connected to that is the question is that your MurJ protein interactor, have you seen whether that fishes out something from E. coli or that does not involve in the native context of third protein?
Okay. The second question, or the first question, I haven't thought about that. It's a good question. We were talking about it over dinner because it's an interesting thing to think about. I think it's, how slide E binds. You can't really see it from this. Let me pull up for time, I skipped over a lot of the stuff talking about slide E and protein E, and I don't have those slides in here. But if we look at it, just from the, I'm going to have to go through all of these to get there, but if you, but now I want the video to go. Can I? All right. Yeah, so if you look, there we go. If you look at how it interacts, this alpha helix that comes across here, on slide E, this IF domain really sort of sits on there in this sort of interesting way. But this alpha helix also makes contacts to MRAY in a pretty specific way. This helix is amphipathic for the most part, but all the hydrophilic parts are facing slide E. The hydrophilic parts are facing MRAY, so it's sort of flipped how you would anticipate it. All of those, I didn't, because I skipped over it. Actually, I think you can see it in the, if I go back here. You know, this is, if you look at it from this orientation, these residues here are all of these completely conserved positive charges. They all interact with negative charges in MRAY. And so there is definitely, you know, something about how those interact with each other that stabilize the overall construct. But protein, or slide E does not touch MRAY at all. So it's really completely mediated by this protein E. Your second question was about MurJ. You know, we express all these in E. coli. We've never seen any. I mean, in the case of protein E, we didn't express slide E, but it's just expressed at a high enough level that it came out with the complex. Right. We haven't seen anything like that.
Got it.
These proteins, these peptides are all so much smaller that this, you know, it'd be unexpected that you would see something more significant. No, I mean, these are all bacterial, so none of them are directly glycosylated, but there could be something interesting going on there. We don't see it, though.
So that was wonderful. So I was looking at so your images of the sigil-mediated lysis of cells. Unlike, say, holon-mediated lysis, which just blows up the cell, you're getting this balloon near mid-cell, suggesting maybe that these are specifically targeting septal peptidoglycan synthesis and not lateral cell wall synthesis. So what's so special about peptidoglycan?
Well, in E. coli, at least, almost all of the peptidoglycans being synthesized at the septal division point. So E. coli grows in that way, where it grows from the center point, and all the new peptidoglycans getting added there. So I think that's why that's sensitive. If you had other, I mean, there are other cells. It's an interesting question to say, what would their phenotypes look like if you had a sigil that you threw into there? You know, would they do something different? They probably would, because there are some that grow from the poles, right? And in those cases, you might see a different story in those cases. Yeah, it's an interesting thing to think about.
Still going strong? Can you handle one more? All right.
I just had a question about the proline that's in protein E. Is it cis or trans conformation? And is that conformation, because slide E is a proline isomerase, so is it influenced by slide E? And does that alter the conformation of protein E such that it binds better to MRAY? Yeah,
no, it's a great question. And in fact, I think it was a misdirect for people for a long time, because people knew that proline was essential, and you had the slide E, so people thought, oh, well, clearly what's happening is it's catalyzing some conformational change in this protein to then do its function. The reality is that we can knock out slide E, we can knock out its proline isomerase activity, and you still get the complex forms, you still get lysis. And so it's not dependent on that proline isomerization. It's a cis, proline. So
it's a cis proline.
Sorry, no, it's a trans proline. I don't know why I said that backwards. It's a trans proline, as virtually almost all prolines are, right? So we're not at a resolution where I could say that 1,000%, but there's no indication that it's a cis.
Have you done molecular dynamics to see the conformational change that would occur if you change it from a trans proline to a cis proline, and would that affect the mechanism of inhibition? Yeah,
I think I doubt it. I mean, these are great questions, so thanks. My guess is that the sort of short answer to that is it's really the kink that role of that proline plays, and so whether it's cis or trans probably doesn't matter that much. We haven't done the MD, so we don't know that explicitly.
Okay, so with that, let's thank Bill for a wonderful talk, educational in more ways than anyone of us could have ever thought. There's an FAES-sponsored coffee reception. Please join us and continue the conversation. Thank you.